Hi, class. Today we're going to talk about aldolase involvement in glycolysis and gluconeogenesis. So aldolase is one of the most important enzymes in the pathway known as gluconeogenesis, which is glucose biosynthesis, and also glycolysis. Aldolase catalyzes the reaction where two 3-carbon molecules combine to make a single 6-carbon molecule. So we're taking two little ones, making one big one together. Aldolase is a key component in glycolysis. In glycolysis pathway, the products and reactants are reversed where a 6-carbon molecule is split into 3-carbon molecules. So, I'm sorry, 2 3-carbon molecules. So for aldolase, we're putting two small ones together. For gluconeogenesis, we're putting two together. For glycolysis, we're splitting two apart. We're going to talk about why these methods are important and how they are utilized in the body. But first, let's talk about aldolase and its function as an enzyme. Then we'll dive into the roles of glycolysis and gluconeogenesis. So aldolase, the genetic coding for the glycolytic enzyme aldolase is found on aldoa gene. It is on chromosome 16. It catalyzes the reversible reaction of fructose 1,6-biphosphate to glyceraldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, DHAP. 3 different genes code for the 3 isosomes of aldolase. We have A, B, and C. Those are just the 3 different isosomes. Recall that isosomes are just different formations of the same materials. These 3 isosomes are expressed differently through development. Certain diseases have been seen to be associated with mutations. Mutations associated with the aldolase include glycogen storage disease, number 12, which is an autosomal recessive disorder associated with hemolytic anemia, and also multiple types of cancer, including lung, renal, and hepatocellular carcinoma. Now, this makes sense because if we are messing with the system for glycogen and the storage of it, it's understandable that we would see some diseases associated with that process in the glycogen storage disease. And also, anytime we see mutations in a gene, it is very common to see carcinomas associated with those. So those add up with the mutations there. Okay, so aldolase is a large enzyme. The active site utilizes 4 amino acids residues. We have 2 lysines, 1 glutamic acid, and 1 arginine. The key catalytic amino acid residuals involved in the reaction are lysine and tyrosine. The main amino acid residue in the aldolase active site is lysine 2,2,3. Other amino acid residues act as a base or acid to help the reaction proceed. Lysine 97 and arginine 295 act as bases, while glutamic acid 178 acts as the acid. Tyrosine acts as an efficient hydrogen acceptor, while the lysine covalently binds and stabilizes the intermediates. Now, let's talk about the first step in aldolase mechanism. We'll be starting with fructose 1,6-biphosphate, which is in the glycolysis pathway. So remember, we're going to take the long molecule, and our ultimate goal is going to be breaking it into 2 smaller molecules. Excuse me. The open ring fructose enters the enzyme and binds to the lysine, with the oxygen taking a hydrogen from the glutamic acid. Next, the arginine takes a hydrogen from lysine, forming a double bond between lysine and carbon, and it kicks off the oxygen, creating water. Then lysine 97 takes the hydrogen from the oxygen, forming a carbon-oxygen double bond, breaking the carbon-to-carbon bond, and forming a new carbon-to-carbon double bond, breaking the one of the carbon-nitrogen bonds. So it's kind of it's not that complex of a system, and you can see parts of it in the pathways on the picture here. We'll look at it a little bit more here in a second. At this point, the first product, glyceraldehyde 3-phosphate, is formed and it leaves the enzyme. So in this process, we're looking to form slightly two different versions of our byproducts here. So if we look at this picture, where's my cursor? On the right here, we talked about, you can see the water on this side forming here. Again, that's when the oxygen comes off, it grabs a hydrogen, 2 hydrogens rather, and forms water in the process. We see that a couple of times in this reaction here. We can also see GAD being formed and DHAP. So this is, it's a little hard to follow, but you can look at it a little bit more closely and you'll see how the different molecules will split off, forming their own products there, just through the process. So in the 4th step, the lysosine forms the nitrogen-carbon double bond, breaking the carbon-to-carbon double bond, and it takes the hydrogen from arginine. Step 5 and 6 are the Schiff's base reversal. They're both a part of that. In the 5th step, water is added, breaking one of the nitrogen-carbon double bonds and adding a carbon-oxygen bond. In the 6th step, oxygen forms a double bond with carbon. The nitrogen to carbon bond is broken and the hydrogen is taken from the oxygen. Finally, the second product, dihydroxyacetone phosphate, is released. The active site can reform by rearranging the hydrogens. So I just down here at the bottom, I put down these, just the main reactant here on the left for this process E, and then we have our products on the right. So we see the fructose 1,6 biphosphate. You can see these 2 phosphates hanging out here on the side in their molecular configuration. And then you can easily see the split between those 2. So these molecules that we're ending up with, the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate, are somewhat similar in appearance. As you can look at them, they have a lot of, you can see the HO up here and the OH down the bottom. The phosphate configurations of the molecular ends look pretty similar as well. So it's easy to kind of track and see how you got from our reactant to our products here. Okay, so let's take a deeper look into glycolysis itself here. Glycolysis, so glyco, which is the sugar, we break down the word, it's easier for us to remember. So glyco means sugar, lysis means break apart. So we know if we're going to be breaking it apart, this has to be the longer one that we're starting with. And basically we're breaking apart the sugar here. So starting with a molecule of glucose, we see it degraded into a series of enzymatic catalyzed reactions. This process will yield 2 molecules of the 3-carbon compound pyruvate. Additionally, free energy is released and utilized in the formation of ATP and NADH. Let's look at where these products go. So glycolysis is the breakdown of glucose. It is used to generate 2 net adenosine triphosphate, which is just ATP, molecules, which provide a readily available source of energy for various reactions within the cell. We also see 2 pyruvate molecules, which can be further converted into lactate used in gluconeogenesis. We'll see acetyl-CoA, which is used in the citric acid cycle. We're going to see oxaloacetate, which is also used in the citric acid cycle. And then we'll see alanine, which is used in the transamination reactions. So the studying of glycolysis actually goes back for a long time. This has been something that researchers has been looking into and trying to understand since the early 1800s and the 1900s. In fact, it was the first metabolic pathway to be elucidated, which was explained way back, again, in the late 1800s, early 1900s. Some mammalian tissue relies solely on glycolysis as a storage for their metabolic energy, and many anaerobic microorganisms rely entirely on glycolysis. So why is this important? Well, our bodies rely on glucose for fuel. We need a constant supply of energy for our bodies to be able to perform at all, much less adequately. And so where does this particular reaction occur? It happens in the cytosol. So we can see in our image here, This is the cytosol within the cell. So this is our cell membrane here. You can see the heads and tails of the cell wall there. This is going to be a mitochondria here on the right. And you can see how we have citric acid cycle in there and oxidative phosphorylation, help you realize what organelle we're looking at there. But out here in the cytosol, we're going to look at glucose, and then we can see glycolysis is going to utilize the ATP there, and we see the NADH on the bottom, and we're creating pyruvate, which will ultimately go to the mitochondria for usage there. But this reaction occurs in like that watery part of the cell, not within an organelle specifically. Okay, so let's look at this diagram here. We're looking at glycolysis. So we're looking at the breakdown of our longer molecule into the smaller molecules. So there's, let me see if I can move this picture here. Okay, so there are a lot of steps here that are kind of squished into here. You can see 1, 2, 3, 4, 5, and then we continue down to 10 for our steps on the right side there. So we're specifically going to hone in on step 4. There are 2 phases of glycolysis. This left side is the preparatory phase. It is made up of the first 5 steps of glycolysis. Energy is consumed to convert glucose into 2,3 carbon sugar phosphates, G3P. So you can see that right here, how energy is consumed. We take ATP, we convert it to ADP because we steal one of those phosphates and it's utilized in the reaction. The enzymes are right here in this teal color. Recall that our enzymes are going to end in ACE, ASE. So we can see right here, there are several different enzymes utilized. We have hexokinase, we have all these different ones here. Here's our aldolase. And if you look at the second side, you'll see a bunch of different enzymes over there as well. So the second side is going to be the payoff phase. It is the second 5 steps of glycolysis. In the payoff phase, we see oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of NADH. The results of the payoff phase are 4 ATP, 2 NADH hydrogen, and 2 pyruvate molecules. As we can see from this image, aldolase is the enzyme utilized specifically though in step 4 of glycolysis. So let's round back over here. Here we see aldolase right here. Here's our enzyme. So we can see this is fructose 1,6-biphosphate, which we've been discussing is the molecule that we're going to break down. And then you can see right here, we have glyceraldehyde 3-phosphate and dihydroxyacetone, phosphate. So we're going to look at that a little bit more in the next image here. But one more thing I wanted to draw your attention to is specifically in the preparatory phase, we are requiring ATP. And in the payoff phase, we're actually producing ATP. So just a couple of differentiations of that process there. Okay, so honing in a little bit more on step 4 of glycolysis. Let's take a closer look here at this top image here. This is the lysis step. So that's the breaking apart step that we talked about before. Just have no good spot for that picture. You can see it breaking apart right there. So fructose 1,6-biphosphate is split into these 2 3-carbon molecules. And they show that nicely in this diagram through the blue and the purple here. I'm sorry, the green and the purple. So right here, it kind of cuts across the center to show you where the purple section is going to be. And then it shows you right there. And then it cuts across the center with the green. And we can see the molecule down here that is formed in the green. So that helps us just kind of track the different atoms and molecules in this process so we can see how that is split into 2 there. So our other image here, let's take a look at standard free energy. So positive, so it is, we see that it is positive here in our G number down here. Here we go. Now recall that a negative G value is going to be spontaneous and a positive G value is non-spontaneous. This is a really low number. So we can probably force this reaction either direction, But why that's important is because recall, we're going to switch this reaction the other way later on. So having that low value right there allows us to go back and forth between which way the reaction occurs. Although aldolase reaction has a strongly positive standard free energy change in the direction of fructose 1,6,5-phosphate cleavage, at the lower concentration of reactants present in cells, the actual free energy change is small, and the aldolase reaction is readily reversible. We're going to take a look again at that later when we get to the second step. Sorry, I'm losing my voice today. Okay. So let's take a look at our other pathway. This is gluconeogenesis. Recall that glycolysis is the breakdown of sugar and occurs in the cytosol. Gluconeogenesis occurs primarily in the liver. And let's break down the word here. So remember, gluco is glucose, neo meaning new, and genesis is like the formation of. So we're forming new glucose. We're making a new molecule with that. So when the body is depleted of glucose and glucogen sources are depleted, it can utilize gluconeogenesis. Recall that gluconeogenesis begins with 2 3-carbon molecules, pyruvate, that combine to make a single 6-carbon molecule, glucose. Although this reaction is the opposite of glycolysis, and the products and reactants are swapped, it is not simple glycolysis in reversed. It does, however, utilize some of the same enzymes, including aldolase. Why can't we just reverse the process of glycolysis for gluconeogenesis? And the answer to that is in standard free energy. While 7 of the reactions are in fact reversible, 3 of the reactions in glycolysis are so highly exergonic that they are more or less irreversible. This is seen in their large negative delta G values. These 3 reactions are catalyzed by hexokinase, PFK1, and pyruvate kinase. Gluconeogenesis uses detours around these reactions. For instance, fructose 1,6-biphosphate instead of fructokinase 1. So let's look at this picture here. So on the left, we have glycolysis. So that's the pathway that we were previously discussing. You can see the hexokinase, all these different enzymes on the left. Here's our aldolase kind of in the middle. And then it comes all the way down, creates our 2 pyruvates. Now, when we're coming the opposite direction, we call that gluconeogenesis. Recall that we can't completely swap the reaction, just action for action, because we have some reactions that are too exergonic to be able to pull that off. So we have a couple of detours. So let's take a look at those here. So right here, we have a detour. We're coming around this way. This one in glycolysis, we see phosphofructokinase 1. And in gluconeogenesis, we see fructose 1,6-biphosphatase 1. So we have to go a slightly different way so that we're able to make these reactions spontaneous so that they will actually occur. Okay. So how is gluconeogenesis affecting the body itself? So gluconeogenesis is regulated by insulin, glucagon, and cortisol. Its main purpose is to maintain blood glucose levels in the fasting state. The balance between stimulatory and inhibitory hormones regulates the rate of gluconeogenesis. So our bodies are able to control when we need to go through this pathway and when it is unnecessary. Many tissues depend primarily on glucose for maintaining adequate energy levels for their proper function during fasting. When we consume food, our bodies will experience a spike in glucose. However, the body will continue to need glucose even when we're not eating. Some organs that require a steady supply of glucose are the brain, erythrocytes, the renal medulla, the lens, the cornea of the eye, the testes, and the skeletal muscle during exercise. The brain in particular requires a steady stream of glucose, accounting for about 70% of the glucose produced by the liver in a fasting state. Gluconeogenesis is regulated by glucagon through changes in allosteric regulation, reduces the levels of fructose 2,6-biphosphate, covalent modifications of enzyme activity, the phosphorylation of pyruvate kinase resulting in its inactivation, and induction of enzyme gene expressed glucagon via CRE response elements increase the expression of PEPCK. For gluconeogenesis to occur, the ADP-ATP ratio must be very low, since gluconeogenesis is an energy-demanding process requiring high-energy molecules to be spent in several steps, which is one of the reasons this isn't necessarily the best way to get glucose, but when it's necessary, it's necessary. Okay, so let's take a look back at the basics to help separate these two in our head. Let's put this over here. Okay, so now that we've discussed these pathways and have a better idea how the processes are related, we'll re-examine why these different processes occur and when they are needed. So a couple of words that we didn't necessarily go over before, but it's important to kind of relate to what we're talking about here. So we have catabolic reaction. So let's look at this first. We have glycolysis versus gluconeogenesis. Glycolysis is on the left. It's kind of this blue teal color. Gluconeogenesis is going to be in our purple. So glycolysis is the set of reactions that convert glucose into 2-pyruvate molecules. We've discussed that quite a bit at this point. It uses the raw material glucose, occurs in the cytoplasm of the cells, whereas gluconeogenesis is going to occur mostly in the liver, a catabolic process. So that the best way for me to always remember catabolic is imagine your cat like walking across the table. It's going to bump into something and break it into a couple pieces, inevitably. That's what always happens. So cat means it's breaking things apart. So the glycolysis is breaking something apart. It is a catabolic process. Where we see on gluconeogenesis is anabolic process. So it is putting things back together. Glycolysis is an exergonic reaction, so it's going to proceed spontaneously. Gluconeogenesis is an endergonic reaction, to the point that, recall, we actually have to take a couple detours around some of those highly exergonic reactions found in glycolysis. So the endergonic reaction is going to require ATP to make that occur. Glycolysis, it occurs through 10 reactions. So remember, we had the 5 in the first half, the 5 in the second half. Gluconeogenesis is 2 essentially irreversible reactions in the glycolysis, 2 or 3, are bypassed by 4 new reactions. So it's actually going to be longer because of just the way that process works. Okay, so let's take a look at our last slide here, just to kind of recap what we've gone over here today. Aldolase is an important enzyme utilized in both glycolysis and gluconeogenesis. It catalyzes the reaction to and from fructose 1,6-biphosphate and dihydrogen acetone phosphate glyceraldehyde 3-phosphate. The pathways between glycolysis and gluconeogenesis are primarily reversed with the addition of a few workaround steps in gluconeogenesis due to, again, those strongly exergonic reactions, making them irreversible. Because the body, specifically certain organs like the brain, need a constant supply of glucose, we have multiple pathways to break down glucose and provide constant fuel for the body. Although both participate in the production of energy, gluconeogenesis is key in maintaining blood glucose levels. So remember, that's the one that we rely on later. So glycolysis is going to happen in the earlier stages following consumption of food as we break that down. Gluconeogenesis is really more something that we rely upon later as our body has processed through that original glucose that we consume, and we are continuing to need that energy input at a later time. Mutations in the gene can lead to these roles being completed improperly. Associated diseases include glycogen storage disease 12, as well as different types of cancers. So again, if we're going to mess up a system that is helping us break down sugars and consume glycolysis or consume glucose at a rate that our body requires it, when there's a mutation in there, it's going to affect that system, not allowing us to properly have glucose when our bodies need it. And of course, we always see cancer as a mutation with these genes, when that's our primary fallback for mutations. Well, I hope you guys were able to track with this today and recall what the main differences are between glycolysis and gluconeogenesis.